Hello everyone, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. And in this particular module, we are trying to understand how the genetic information is flowing from one generation to the next generations. In this context, so far what we have discussed about the law of inheritance. So in that context, we have discussed about the Mendel's first law of dominance. When we also discussed about the Mendel's second law, and in the previous lecture we have also discussed about the Mendel's third law. Mendel's first law is mostly been related to the genetics of the inheritance of the single gene, whereas in the previous lecture we have also discussed about the inheritance of the two gene or the polygenic situations. So far what we have discussed that the factors which the term the Mendel has pointed that the factor is responsible for carrying the genetic information from the one generation to the next generation. So there are instances where actually the external factors which were not the part of the genetic material is also influencing the phenotype, and there are many cases where the people are actually having the identical genotype, but they were showing the different types of phenotypes depending upon the external factors. Now, it's these external factors, the influence of external factor onto the phenotypic characters is a very interesting topic to understand, right? So in today's lecture, we are initially going to discuss about how the external factors are controlling or regulating the phenotypic characters. And then subsequent to that, we are going to discuss about the sex determinations. We are also going to discuss about the genetic disorders. So let's start the today's lecture. So the first thing what we would like to understand is how the external factors which are not the part of the cell, which are not part of the DNA, the genetic material, right, is also influencing the genetic factor or also influencing the phenotypic outcomes. But until the people who are following the Mendelian genetics, people understand the one concept that the genotype is actually governing the phenotype, right? But then there was a concept of the twins, right? So people have found that the twins brother, right? If there are twins, one is in the X conditions, the other one is in the Y condition, and if they continue to grow in those different conditions, what we have found that these two particular individuals, which are genotypically identical, are actually having the lot of variations in their phenotype. And then people have started discussing about the how these kind of changes are happening, and all these genetic changes or all these phenotypic changes are actually the part of epigenetics. So the C.H. Weddington, the person, coined the term epigenetics in the year of 1940 to describe how the environmental influence on the developmental event can affect the phenotype of an individual. Now, this is a very interesting observation that I have just given you an example, right? If you have a twin, right, and suppose you have the zygotic twins, for example, not the Siamese twins. So if you have a zygotic twins, and when you if you keep one twin in the hot weather, right, for example, and the other one you keep it into the cold weather, right, and what you see here is at the end the phenotypically they are actually going to be different, right? So this kind of changes where the DNA, because these are the zygotic twins, so DNA or the genetic material is same, but the guy who is staying in the hot weather or the guy who is staying in the cold weather are actually phenotypically very different in terms of many aspects, in terms of, and not only that, right, this is the environmental influence, whereas there are other factors also which are actually influencing the overall phenotype, right? So it's not like they are very different in terms of like the skin color only, right? They are also different in terms of their metabolism. They are also very different in terms of many other phenotypic characters. Now, how this all this started? This is started with the study of the genetics in the year of 1871. So there will be a development of a field which is called as genetics, and then the epigenetics, which is the term is coined by in the year of 1942, and then the year of 1948. The DNA modifications were discovered. So one of the modification was the DNA is getting the methylated. Then in the year of 1951, the Barbara and McClintock actually discovered the nature of the mutatable locus, right? And then in the, the DNA methylation and gene switching is being discovered in the year of 1975. And then in the year of 1984, the role of parental and maternal genome is being discovered in the development. And then they've also started doing a lot of experiment where they are actually cloning and doing all this kind of experiment, and as a result of that, in the year of 1996 it has discovered the dolly, which is dolly being cloned. And then the subsequent to that, people have also studied related to the histones. So remember when we were discussing about the genetic material of the organisms and how the packaging is happening and all that, we discussed that the histone is very, very important. And histone is actually controlling the packaging of the DNA, right? So if you control the packaging of the DNA, although the two individuals will be of the same genetic material, their expression status is going to be changed because you are actually changing the histones by doing the different types of modifications. So what are the different types of traits which are related to the epigenetics? Okay. So epigenetic traits are stable, mitotically and meiotically heritable phenotypes that result from the change in the gene expression without alteration into the DNA sequence. So this is a very, very, very interesting thing, right? So basically what you have is you have the, for example, you are actually going to have the epigenetic tags, right? So you are actually going to have the epigenetic sequences, and you are going to have the epigenetic tags onto the female eggs. You are going to have these tags onto the sperms, and then when they are actually going to join, they are actually going to form the eggs, right? And they are actually going to form the embryo. So that's how the epigenetic tags from the male and the female are actually going to come together, and that's how they are actually going to be resulted into phenotypic changes into the, because the reproductive cell is going to grow and give you the complete organisms, right? And since these epigenetic tags are responding to the environmental factors, they are actually going to give you the different types of individuals. So the epigenetics is the study of the way in which the epigenetic traits are altering the cells and the tissue specific pattern onto the gene expression. So basically epigenetics is the study how the DNA, the information from the DNA is actually going to be utilized for production of the protein. So basically it is not changing the DNA, but it is changing the protein. And remember that the protein is actually the final molecule which is actually going to be responsible for providing the phenotype, right? So so if you alter the protein, if you reduce the amount of protein, if you increase the amount of protein, that's how you are actually going to change the phenotype, right? And that all is being done by the many types of epigenetic traits and many types of epigenetic characters. So how the regulation of the epigenetics works? So the gene expression uses the reversible modification of the DNA and the chromatin structure to mediate the interaction of the genome with a variety of environmental factors and generate changes in the pattern of gene expression in response to these factors. So remember that the epigenetics is a very, very big topic, and epigenetic regulation is also very important, and where you are actually having the different types of mechanisms, right, where you can actually be able to modify the DNA, you can actually be able to do the some kind of related to the DNA packaging and so on. So epigenetic regulation is itself is a very, very important topic, and in this particular lecture, what we are trying to do is we are actually going to introduce you to the topic so that if you find it more interesting, you can be able to study, right? Then the concept of the epigenome. So the epigenetic state of a cell, the organism has one genome, and genome can be modified in a diverse cell type at a different times to produce the many epigenomes. So remember that the genome of an organism is same, right? Whether it is the genome present in the liver, whether the genome is present in brain, whether the genome is present in our eyes, tongues, and so on, right? Or it the genome what is present in the pancreas and so on. But you see that the protein production from all these organisms are, or all these organs are very different, right? And that all is being done because genome is being modified in a different way, and that's how it is producing the epigenome, right? Which means the genome which is being modified by the epigenetic factors, right? And that's how it is actually going to be changed, right? Now, what is epigenome? So, epigenome is actually the way in which the normal genome is being altered, and that's how it is actually going to be called as epigenome, right? So, there are different types of alteration. So, alteration is you are actually going to have the epigenetic repressors, or you are actually going to have the epigenetic activators or the modelers, right? Epigenetic repressors are actually going to down regulate the expression of certain genes, so it will actually going to reduce the amount of that particular protein, whereas in the case of epigenetic activators or the remodelers is actually going to increase the amount of that particular protein. And you know that when we were discussing about the polygenic inheritance, what we said is that it is not the single gene which is actually going to regulate a particular type of phenotype. Sometimes what phenotype you will observe is being regulated by the multiple genes, right? And when the multiple genes are producing, the actual phenotype is the summation of the effect of the individual protein, right? Classical example is the skin color, right? So skin is being controlled by not by one gene, right? Skin is being controlled by multiple genes. And if the multiple genes are being responsible for the skin color, what you can do is you can actually be able to modulate the production of these proteins, and that's how you can be able to have the different shades of a particular skin, right? So you can have the no color, right? So then it's going to be very white skin. If you have the expression of all the genes, and then it is going to be a black skin, right? So all the epigenetics alteration is encoded within the genome, and that's how it is actually going to be carry from one generation to the next generations, and that's how it is actually going to be responsible for the final outcomes, although there's no change into the DNA content. Now, talking about the epigenetic alterations, so the epigenetic or epigenome can be transmitted to the daughter cell via the mitosis and the future generation via the meiosis. So remember, all these epigenetic tags are going to carry in a particular family, and that epigenetic tag will carry from next one generation to the next generations depending upon the environmental factors. These epigenetic tags are actually going to be expressed more or less, and as a result you might see that some of the people who are staying in one genetic environmental conditions versus the next environmental conditions, there will be a alteration of the phenotypic change. So there are three major mechanisms which actually been responsible for the epigenetic outcomes. First is the reversible modification of DNA by addition or removal of the methyl group. So this means there will be a methylation, right? And so if there will be a methylation of the DNA of the different sites, methylation is going to modulate the gene expression, right? And there is a developed mechanism through which the methylation is actually going to change the gene expression. And when there will be a change of gene expression, it is actually going to change the amount of protein what is going to be produced, and as a result you are actually going to show the one phenotype versus the second phenotype, right? The same is true for even the eye color also, right? So if the amount of protein is actually going to decide what color grade you are actually going to exhibit into your eyes. Then the second is the alteration of the chromatin by addition or removal of chemical groups to the histone proteins. So remember that the histones are packed with the DNA by a positive negative interaction, right? So histones are positively charged proteins, right? These are histones, right? When we were discussing about the nucleosome, that time we discussed, right? So histones are actually going to cover the DNA, right? And this DNA, what is present onto the histone core, will not be allowed to participate into any of the downstream reactions, like for example replication, transcription, and so on. So if you are actually going to make the epigenetic changes onto these histone proteins, because of which if they will lose their positive charge, right? For example, if you do the acetylation, for example, if you do the acetylation, so it is actually going to impart the negative charges, right? So if you do the acetylation, basically you are actually going to reduce the effective positive charges. You are actually going to bring more negative charges onto the histone proteins, and as a result of that, the DNA what is being rolled onto this is going to be more freely available for the participating into the different types of reactions. For example, it is going to be participate into the transcription, and as a result of this portion of DNA what is being not bound to the histone will actually going to be get transcribed better, and that's how it is actually going to give you the protein corresponding to this region, and because of this protein it is actually going to result into the phenotypic changes. Then you are actually going to have the third aspect where you are going to have a regulation of gene expression by the non-coding messenger RNA or the miRNA, right? So these are some of the three mechanisms which people have proposed for observed epigenetic changes into the particular organism. And what is the role of these epigenetic changes? So they are actually going to have a role on to the imprinting. They are also having a role into the development of cancers. They are also having a role into the behavior of an organism, and they are also going to have a change onto the environment genome interaction, right? Moving on to the next topic, and the next topic is the sex determination in the organism. So sex determinations always been controlled by the sex chromosome, right? So sexual reproduction is the formation of offspring that are genetically distinct from their parent. Most often two parents contribute genes to their offspring, right? Among the most eukaryotes, sexual reproduction consists of two processes that led to the alteration of the haploid and diploid cells. The meiosis reduces the haploid gamete, and fertilization produces the diploid zygote. The term sex refers to the sexual phenotype. Most organisms only have two sexual phenotypes, that is the male and the female. The fundamental difference between the male and female is the gamete size. The male produces the small gamete, which has been called as sperm in the case of humans, and the female produces the relatively large gamete, which has been called as egg. The mechanism by which the sex is established is a sex determination. So we have the different types of mechanisms. You have the XX XO sex determinations. So mechanism of sex determination is the in a grasshopper studied by the MacLean is one of the simplest mechanism of the chromosomal sex determination. It's called as XX XO system. In this system, the females have two X chromosome and the male possesses a single X chromosome XO. There is no O chromosome. The letter O signifies the absence of a sex chromosome. In meiosis in female, the two X chromosomes pairs and then separate with the one X chromosome entering each haploid. So basically you have the female, you have the male, the female is XX and the male is X only, right? And in the male, the single X chromosome segregate and meiosis to half the cells and the other half is no X chromosome, right? So basically it is going to give you the two gametes. One is X, the other one is X, and in this case you are going to have the two gametes. One is X, the other one is actually going to be O, which means minus, right? So there will be XO, right? Now when there will be a fertilization, so if the X is going with X, right, then it is going to produce the XX. If the X is going with the X, it is going to produce the XX, right? And if the X is going with O, then it is going to produce the XO, and if with this one also you are going to produce the XO, right? So because the male produces two different gametes with respect to the sex chromosome, they are said to be heterogametic sex. Females, when we produce the gamete, are full with respect to the sex chromosome and the homogametic sector. So in the X-sex exosystem, the sex of an individual is therefore determined by which type of male gamete fertilizes the egg. X-bearing sperm unite with the X-bearing to produce the XX zygote, which then develop into a female, right? So remember that if the XX is being formed, then it is going to be female. If XO is being formed, then it is going to be male, right? So, and if the sperm lacking X chromosome and the X bearing X to produce the exozygote, then it will going to develop into the female. Then the second way of character is the XX and XY. So this is the most popular one, which is actually happening in many type of organisms, including the humans, right? So where you are actually going to have the females are actually going to have XX, and the male are actually going to have the XY. So this is the female, right? And this is going to be male, right? And one after the meiosis, you are actually going to produce the eggs, which are going to have X, or you are going to produce the sperm, which is going to be either X, or you are going to produce the sperm, which is, so this is going to be egg, this is going to be sperm, and when there will be after fertilization, if you are producing XX or you are producing the XY, right? So you basically have 2X and, right? So if the X is combining with X, you're going to have XX. If the X is combining with Y, then you're going to have XY. If the X is combining with X, you're going to have XX. And if the X is combining with Y, you're going to have the XY, right? Now, this XX is going to be female and the XY is going to be male, right? So here again, the male is actually going to decide what of the sex of the offspring, right? So the female is homogametic and the male is heterogametic. Male is heterogametic, so it is going to have XY. Female is going to be XX, right? And ultimately it is going to be so humans like Drosophila have XXXY determination, but in humans the presence of a gene on the Y-axis must determine the maleness, right? And this is we are going to study further, where we are going to talk about the Y-linked characters and so on, right? Then the sex chromosome do not segregate properly in meiosis or mitosis, illustrate the importance of Y chromosome in the human sex determinations. The XY determinations are not generally homologous. They do pair and segregate into the different cells in the meiosis. Now, so this is what it's showing, right? So in the parent generation, you have the XY and the XX, which is going to be male and female. When they will be going to be meiosis, then it is going to form the gametes containing X, gametes containing Y, or gametes containing XX, right? And then there will be a fertilization, then it is going to produce the XX or the XY, right? And that's how the inheritance of a sex in an organism with XY chromosome results in the equal number of male and the female offsprings. Then we have a third system. Third system is ZZ and ZW. So in this particular system, the female is heterogametic, which means the ZW is actually for the female and ZZ is actually for the male, right? And the male is homogametic. So to prevent confusion with the XX and XY system, the sex chromosome in this systems are labeled as Z and Y, but the chromosome do not resemble as ZS and WS, right? So female in this system are ZW. After meiosis, half of the egg have a Z and the other half are actually having the W. Males are actually ZZ, right? So all sperms containing only the Z chromosomes, and it's found in birds, moth, some amphibians, and the fishes. Then we also have the sex determination into the honeybee, so which is a haplodiploid system. So in some insect in the like Hymenoptera have no sex chromosome. Instead, the sex is based on the number of chromosome sets found in the nucleus of the each cell. So male develop from the unfertilized eggs and the female develop from the fertilized eggs. The cells of the male Hymenoptera possesses only a single set of chromosomes, right? That is means they are haploid. Whereas the cells of the female possesses the two set of chromosomes, which means it is going to be fertilized and they are diploid. So the haploid diploid method of sex determination produces some odd genetic relationship. When both parents are diploid, siblings on the average have half their genes in common because they have a 50% chance of receiving the same alleles from the each parent. In these insects, the male produces sperms by mitosis, so all offspring receive the same set of paternal genes, right? So there will be no diversity and there will be no deviations. The diploid female produce eggs by the normal meiosis. Therefore, the sisters have a 50% chance of receiving the same allele from their mother and a 100% chance of receiving the same allele from their father. The average relationship between the females is 75%. Brothers have a 50% chance of receiving the same copy of each of their mother's two alleles and at any particular locus, so their average headness is only 50%. The greater genetic relatedness among female siblings in insects with the haploid-diploid sex determination may contribute to the high degree of social cooperation that exists among the females of these insects. So this is what exactly is shown here, right? Now, once we discuss about the epigenetic contributions to the genetic variations, then we talk about the sex determinations. Now let's move to the next topic, and the next topic is if there will be a alteration into the DNA, then it will be result into the different types of disorders, right? Some of the disorders could be related to the genetics, right? Some of the alterations may not be noticed because that may not be causing the significant alterations into the phenotype and so on. So let's talk about the genetic disorders. So we have the two different types of genetic disorders. The genetic disorders where the particular type of chromosome is being altered and the disorders which are going to follow the Mendelian inheritance. So the first is X-linked recessive traits. So these X-linked recessive traits have a distinct pattern of inheritance. They are actually appear more frequently into the male because the male needs inherited only a single copy of allele to display the trait, whereas the female must inherit two copies of the allele, one from each parent to be get affected. So this is actually a characteristic of the X-linked recessive traits, right? Where the males are going to be get affected even if they are actually getting the one copy of X chromosome, whereas the females are only affecting if they are getting the affected X from both the parents, right? So it is actually going to be more problem for the male. So all the X-linked recessive traits would be a problem for the males rather than the females. So even if the male are getting one X chromosome where this particular trait is present, they are actually going to exhibit the particular disease. So this is what is shown here, right? So if you have the female, right? So which is where the female is actually a carrier. So it is actually having the gene, but it's only having the only one X is being carrying the wrong information. The other X is perfectly fine. So it is actually going to be a carrier. One, this is going to mate with the normal male. So normal male is going to be like this, right? And then they are actually going to produce the male, right? And the male is only going to have one X chromosome, which is from the female side. So it is carrier. And that's how it is actually going to have the XY. And as soon as they have the one X, which is from the affected gene, carrying the affected gene, it is going to show you the disease actually, right? So wherever there will be, whereas in the case of female, it's only going to show the problem when this particular male is also going to go for the sexual reproduction with the carrier females or with the affected females, right? Then only it is actually going to show you the this is what is shown here, right? So the X-linked recessive traits appear more often in males and not from the father to son. So it doesn't go from father to son actually, because from the father to son, the son is going to get the X from the mothers, not from the father, right? It's only going to get the Y character. And daughters are also not going to get as long as the mother is not having the affected X chromosome. So only carrying mother, the mother for the carriers of this particular trait, and the father is having the disease, then only there is a chance that the son and the daughters are actually also going to get the X-linked characters. One classical example is hemophilia. So X-linked recessive trait in hemophilia is hemophilia A, also called as the classical hemophilia. And in this particular disease, there will be absence of protein necessary for the blood to clot, right? So remember, there's a cascade of reactions, what is required for the blood clotting, and one of the protein is missing, and that has been responsible for development of hemophilia, right? So the factor 8 is located onto the tip of the long arm of the X chromosome. So hemophilia A is an X-linked recessive disorder. People with hemophilia A bleed excessively. Even small cuts and bruises can be life-threatening, right? Then we have another disease which is called as color blindness. So the color blindness is also X-linked recessive traits, and it is a color blindness, right? So when the human eye, color is perceived in light sensing cone that link the, so each cone contain one set of three pigments, right? So you have the light absorbing pigments and so on, right? And human actually detect only three colors: red, green, and blue. But the brain mixes the signal from the different cones to create a wide spectrum of color that we perceive, right? So these are actually the basic color, right? And each of these pigment is encoded by a separate locus. The locus for the blue pigment is found onto the chromosome number 7, and for the green and the red, it is like close to the onto the X chromosome. So the most common type of human red blood color blindness are caused by the defect of the red and green pigment, and we will refer to those conditions as red green color blindness, because the red and green pigments, the region which is coded for the red and green pigment, like very close to onto the X chromosome. So mutations that produce the defective color vision are generally recessive, and because the genes coding for the red and green pigment can be located onto the X chromosome, it is actually going to follow the X-linked recessive characteristics. So this is what it's shown here, right? You have the normal vision, color vision for the female, then you have a color blind male, and when they will come together, there will be a fertilization. So in the into the first generations, and then you are going to have the normal color vision females, which are actually going to carry the the X chromosomes even from the father, right? Whereas in this case, the normal color vision male, because it is going to follow, right? So normal color vision male actually. So the red-green color blindness is inherited on X-linked recessive traits in humans. So the normal female and the color blind male actually. So in the reciprocal cross between a color-blind woman and a man with the normal color vision, the woman produces only X-bearing gametes. The man produces the gametes which are X and the Y chromosomes, and men inherited the X chromosomes their mother, because both of the mothers X chromosomes bear the XC allele, and all the male offspring will be colorblind, right? So in contrast, the female inherited an X chromosome from the both parents. Thus, the female offspring of this reciprocal cross will be heterozygous, and it will be of normal vision. So, females are colorblind only when the colorblind alleles have been inherited from the both the parent, and that is very, very important. So, X-linked characters are mostly be a problem for the males rather than the females. Then we have the X-linked dominant traits. So X-linked dominant traits appears in the male and female, although they often affect more female than the males. So as the X-linked recessive traits, the male inherited X-linked dominant traits only from his mother. The trait is not passed from the father to son, right? A female, on the other hand, inherited an X chromosome from both her mother and the father, so the female can receive X-linked traits from the either parents. Each children with an X-linked dominant trait must have affected parents, like unless the child possesses a new mutation or the genes, right? X-linked dominant traits do not skip generations. Affected men pass the traits on to all their daughters and none of their sons, as is seen in the children of 1:1, right? In contrast, the affected woman passes the traits on to the half of their son and half of their daughter, as seen, right? So this is what exactly shown here, right? So this is the affected male, and this is the female and female, and then when there will be the second generations, all the all the females are actually going to be affected, and so on, right? So X-linked dominant traits affect both in male and female, and an effect of male have a must have an affected mother, right? Then we have the another disease which is called as the hypophosphatemia. Hypophosphatemia is a disease. Uh, so it's an X-linked dominant trait, which where you have the hypophosphatemia, also called as familial vitamin D resistant rickets. People with these traits have a feature that superficially resemble those produced by the rickets, like the bone deformities, stiff spines and joints, bowed legs, and the mild growth deficiencies. With this disorder is resistant to treatment with the vitamin D, which normally cures rickets. So X-linked hypophosphatemia results from the defective transport of phosphate, especially in the cells of the kidney. People with this disorder excrete large amount of phosphate in their urine, resulting in the low level of phosphate in the blood and reduced deposition of the mineral into the bone. As is common with the X-linked dominant traits, males with the hypophosphatemia are often more severely affected than the females. Then we have the Y-linked traits, a trait resulting from a mutant gene that is carried onto the Y chromosome but has no counterpart on the X is called as the Y-linked or the Hollandric traits, right? Such traits should be easily recognizable because every son of an affected male should have the trait. So female should not ever express it, right? Only the males are affected and the trait is passed from the father to son. So this is the pedigree of the Y-linked characters. So if the father is affected, it is going to transmit all into the son, right? All the time, right? So it's going to go into the son only because it is been a trait which is present on the Y, and the Y is only present into the male. One such example is the hairy heirs, right? A possible example of Y-linked inheritance is the hairy heir trait, in which the bristle hair of a typical length grow from the ear. This trait is common in the part of India and some other population also exhibit. Although the trait show father to son inheritance, there is no doubt that it is complex phenotype. Many of the collectively pedigree can be interpreted in other ways, such as the autosomal inheritance. Then we have the autosomal recessive traits. So autosomal recessive traits normally appear with the equal frequency in both the sexes and appears only when a person inherited two alleles for the trait, one from each parent. So, and if the trait is uncommon, most people carrying the alleles are heterozygous and unaffected. Consequently, the traits appear to the skip generation. Frequently, a recessive allele may be passed for a number of generations without the trait appear in a pedigree, right? So this is the example of the recessive traits, right? So where you know the female are actually getting affected, right, and a male is getting affected. So autosomal recessive traits normally appear with the equal frequency in both the sexes and seems to be skip the generation. So remember that in this generation, it is affecting, and then it is skipping the first cousins, and then again it is skipping and all that, right? One such example is the sickle cell anemia, right? So sickle cell anemia is a genetic disease affecting the hemoglobin, the oxygen carrying protein. So sickle cell anemia was just first described in the year of 1910, and this is a condition of low oxygen tension. Red blood cells when people have the disease lose their characteristic disc shape and assume the shape of a sickle. This is a sickle cell anemia, right? So these are the disc shaped RBCs when they convert into the sickle cell phenotype. The sickle red eye are fragile and break easily, resulting into the anemia. Sickle cells are not as flexible as the normal cells, and therefore it tend to clog capillaries rather than the squeeze through them. And as a result, the blood circulation is impaired and tissue becomes deprived of oxygen. So these are just details of the sickle cell anemia, where you are actually having the mutations into the hemoglobin structure, and as a result of that, it is actually going to change the morphology of the RBCs. Then the second is the thalassemia. So thalassemia is an inherited disease of faulty synthesis of hemoglobin. The name is derived from a Greek word thalassa, which means the sea. And thalassemia consists of a group of disorders that may range from a barely detectable abnormality of blood to a severe or fatal anemia. So the adult hemoglobin is composed of 2 alpha and 2 beta, right? There are two copies of hemoglobin alpha genes, HbA1 and HbA2, which encode the alpha chain, and both genes are located on the chromosome number 16. The hemoglobin beta chain encodes the beta chain, and it is located onto the chromosome number 11. In alpha thalassemia, there is a deficient synthesis of the alpha chain and the excessive change of beta chain, which binds the oxygen poorly, leading to a low concentration of oxygen in the tissue, which is called as hypoxia. Whereas in the case of beta thalassemia, there is a lack of beta chain, right? So basically, there will be a the underproduction of the alpha chain or to the beta chain. Then the second disease is called as the phenylketonuria, and phenylketonuria is the inherited error of metabolism caused by a deficiency into the enzyme phenylalanine hydroxylase. Loss of this enzyme results into the mental retardations, organ damage, and unusual posture, and can result into a case of maternal PQ, severely compromised pregnancy. The classical phenylketonuria is an autosomal recessive disorder caused by the mutation in the both alleles of the gene for the phenylalanine hydroxylase found into the chromosome number 12. So in some cases, the mutations into this particular gene results in a mild form of phenylketonuria, which is called as the phenylalemia, right? Both diseases are the result of a variety of mutation into the pH locus, in these cases where a patient is heterozygous for the two mutations of the pH. Then we also have the autosomal dominant traits. So autosomal dominant traits appear in both the sexes with the equal frequency, and the both sexes are capable of transmitting their traits to their offsprings. Every person with the dominant traits must inherit the alleles from at least one parent, whereas the autosomal dominant traits therefore do not escape the generation. Then one of the classical example is the hypercholesteremia, right? So the one trait usually considered to be autosomal or dominant is the familial hypercholesteremia, an inherited disease in which the blood cholesterol is greatly elevated owing to a defect into the cholesterol transport, right? So cholesterol transport is an essential component where the cholesterol is being transported from the blood to the liver, and that's how it is going to be stored, or the blood is going to be transported from the blood to the adipose tissues, right? So because the cholesterol is a lipid, right, it is not readily soluble in the blood, right? So as a result, it is being formed as a the LDL particles and all that, right? So therefore, it is being transported through the body in a small soluble particle, which is called as the lipoprotein or the LDL, right? And the LDL particles are consist of a core lipid surrounded by a shell of the charged phospholipid and protein that dissolve easily into the blood. One of the principal lipoprotein is the LDL or low density lipoprotein. So when the LDL molecule reaches the cell, it attaches to the LDL receptor and then it is being taken up, right? This is we have discussed when we were talking about the transport of the material into the cell, right? And then you also going to have the hypercholesteremia, because there will be a problem into the alterations of the uptake actually. So there will be a problem into the gene which will code for the LDL receptor. Then the second series of the genetic disorder is where you are actually going to have the problem onto the chromosome, right? So chromosomal aberrations, right? So the changes in the genome involving the chromosome part, where the chromosome or the whole chromosome are being altered, right? So chromosomal aberration or the chromosome mutations. So it is of two types: the structural alterations or the numerical alterations. In the structural alterations, there will be a change of a specific part of the chromosome. So it could be a duplications, deletions, inversions, or the translocations. So what is the duplications? So duplication, the chromosome duplication is a mutation in which the part of the chromosome has been doubled. Consider a chromosome with a segment A, B, C, D, E, F, in which they represent the centromere, right? And then there will be a tandem duplication, there will be a displaced duplication, and there will be a reverse duplication. So in a duplication, in a tandem duplication, the duplication might include the EF segment, right? So this is actually going to have this segment, right? Giving rise to a chromosome with a segment like A, B, C, D, E, F, E, F, G, right? So this actually, get doubled, right? And because of that, you are going to have a chromosome which is like this, and this is actually the centromere, right? Then we have the displaced duplication. So if the displaced duplication is located some distance from the original segment, either on the same chromosome or in a different one, then it is called a displaced duplications. For example, there will be like AB, then CD, EF, GH, and then EF. EF at the end of the particular chromosome, right? So then there will be a displaced duplication. And then you have a reverse duplication. Reverse duplication means the EF is actually going to present as FE, right? So in a duplication can either be in the same orientation of the same signal, or in the preceding examples would be inverted, right? For example, A, B, C, D, E, F, and then F, E, and G. So when the duplication is inverted, it is called as reverse duplication. Then we have a deletion. So deletion could be a loss of the segment of the genetic material, and this is called deletions. So it could be a terminal deletions, or it could be intercalary deletions. So in a terminal deletions like A, B, C, D, E, F, G, the G is going to be deleted, right? So this is going to be your mutated chromosome. And then you have intercalary deletions. So you are going to have the A, B, C, D, E, F, and in between there will be a deletion. So it is actually going to have the A, B, C, E, F, G, right? Then we have the inversions. So it is another chromosomal abnormality in which the order of a gene in a chromosome segment is reversed by an angle of 180 degrees. So remember that we have the genes which are being placed onto the chromosome, right? Remember that the chromosome has a centromere, and then you have a chromosome like this, right? So genes are placed like this, right? Genes are placed like this, right? And they are actually in a order, right? So remember that if the order is like A, B, C, D, E, F, G, H, right? And due to the abbreviation, the sequence genes become like A, B, C, D, G, F, E, and H, right? So there is a alteration here, right? So it could be pericentric inversions. So in the pericentric inversion, the inverted segment of the chromosome contain the centromere. Sometimes it is responsible for the evolution of the organism. For example, you have the A, B, C, D, E, F, G, then it becomes A, E, right? So it is this one comes here and this one goes here, and then you have a D, C, B, F, G. So if you have a pericentric inversion, then the things will go from here to here, and as a result, it is going to have the complete changes. Then we have the pericentric inversion. So in the pericentric inversion, the inverted segment of the chromosome has no centromere. So for example, A, B, C, D, E, F, G, then you have the A, B, C, D, and here there is the gene. So this pigment is actually getting flipped, right? So it is going to be flipped like this, and because of that, the genes which are present on this side will come on this side, and the genes which are present on this side will come on this side. So that will be called as paracentric inversion. Then you have a translocation. So this is a kind of a chromosomal abnormality in which the interchange of the chromosome segment occurs, right? So it could be a reciprocal translocations, right? So when the translocation occurs between the two non-homologous chromosomes, then it is called as reciprocal translocation or the illegitimate crossover, right? Uh, in a heterozygous translocation, one member of each pair of chromosome is normal and the other member is with the interchange segment. Then the non-reciprocal translocation. So in a non-reciprocal translocation, the genetic material moves from the one chromosome to another without any reciprocal change. So for example, if you have the two non-homologous chromosomes like A, B, C, D, E, F, G, and M, N, O, P, Q, R, S, and if there will be a non-reciprocal movement, then there will be some movement of the molecule between the A, B, C, D, G. So you basically have the EF, which is moved from this chromosome to this chromosome, and as a result of this chromosome got changed like A, B, C, D, G, and this one is going to change to M, N, O, P, E, F, Q, R, S, right? Then we have the Robertson translocation. So the long arm of the two epicentric becomes joined to become a single common centromere through a translocation, right? So this is the example of the Robertson translocations. Then we also have the numerical chromosomal variations, right? So where the number of chromosomes are actually being changed. So it is also known as, so the alteration in the number of chromosomes from the diploid set is called as the numerical chromosomal alterations. This is also known as the ploidy. So it could be change in the number of individual chromosomes, then it will be aneuploidy, or it will be change in the number of chromosome cells, then it will be called as the polyploidy. So in the aneuploidy can be because of the loss of the centromere, prevent the spindle fiber from attaching, or the small chromosome generated by the Robertson translocation may be lost into the mitosis and the meiosis. And the third, aneuploidy may arise because of the failure of a homologous chromosome or to segregate or separate into the meiosis or the mitosis. Aneuploidy of two types, hypoploidy and the hyperploidy. So in the hypoploidy, decrease in one or two chromosomes for the diploid set is described as the hypoploidy. So there are two types of hypoploidy, monosomy and the nullisomy. So in the monosomy is due to the loss of chromosome from the diploid set, for example 2n minus 1, right? Remember that all the chromosomes are present as the 2n number, right? So if there will be a loss of one chromosome, then it will be 2n minus 1, and that will be a part of monosomy. Whereas in the nullisomy is a condition in which a pair of homologous chromosome is lost, that means 2n minus 2. So basically it will still be 2n, but there is a loss of two molecules, right, instead of one molecule. So here there will be a one chromosome which will remain unpaired, whereas in this case there will be both the chromosomes are being removed, so it is actually not going to have any chromosome which is remain unpaired. Then we have the hyperploidy. So hyperploidy is addition of one or two chromosome in a diploid set, so it is result into the hyperploidy. So it could be trisomy, it could be tetrasomy, it could be Turner syndrome and a Klippfinger syndrome, right? And the most common is the autosomal aneuploidy, which is a trisomy of 21, which is also called as the Down syndromes. Then we have the polyploidy, right? So addition of one or two set of chromosome to the diploid set result into the polyploidy, and it is commonly noticed into the plant and rare into the animals. There are two kinds, autopolyploidy and the allopolyploidy. So the autopolyploidy, the addition of one or more haploid set of its own genome in the organism results into the autopolyploidy. For example, the watermelon, grapes, bananas are autotriploids, whereas the apple is a autotetraploids. Then you have the allopolyploidy, increase in one or more haploid set of chromosomes from the two different species results into the allopolyploidy. Then triticale is the first man-made cereal. It is obtained by crossing a wheat triticum and a rye sickle cereal, right? So the F1 hybrid is sterile, then the chromosome number is diploid using the colchicine and becomes the hexaploidy. So there are many examples, there are many disease, the important disease which are happening because of these chromosomal numerical alterations, such as the Down syndromes, such as the different types of chromosomal problems. Okay, so what we have discussed about the epigenetic effect of the environmental factors onto the alteration into the phenotypic characters of an organism. So we discussed about the mechanism part, how the epigenetic sites are being carried from one generation to the next generation, and that's how these epigenetic effects are continuing and being carried out from generation to generation. And there are three different types of mechanism which are being responsible for the epigenetic mechanism, in which one is where you are actually altering the DNA by doing the methylations. The second is you are actually changing the DNA packaging, and the third where you are actually using the small non-coding messenger RNAs. Now, these epigenetic control is very, very important, and they actually been used probably by the organism to go for the adaptation into the different types of environmental factors or different types of environmental conditions. Apart from that, we also discuss about the sex chromosomes. We have discussed about the sex determinations into the different types of organisms. So we have discussed about the XX and XO type of sex determination. Then we also discuss about the XX and XY type of sex determinations and so on. And the lastly, we have also discussed about the different types of genetic disorders. So we discuss about genetic disorder due to the the different types of chromosomal linked genetic disorders. So we discussed about the X-linked chromosomal genetic disorders, Y-linked chromosomal genetic disorders, and then lastly we have also discussed about how the genetic disorder arises when there will be a change in the number of chromosome present in a particular organism. So with this brief discussion about the different types of principles and the rules required for carrying the information from the one generation to the next generations, and how we can be able to use this information to understand the uh, phenotypic characters going from one generation to the next generations. I would like to conclude my lecture here. In our subsequent lecture, we are going to discuss some more aspects related to this particular topic. Thank you.